Yeah. Hi. Good evening, everyone. Yeah. Sorry about the disturbance. Just a second. I'll share the screen again. Right. Is my screen visible to everyone?
Yes, ma'am.
Thank you.
Yes,
Thank you
ma'am.
for the confirmation. Thanks. Okay. Yeah. Hi. Welcome, everyone. Thank you for joining this problem solving session 2. So as you know, this is a course which is offered by NPTEL, and it is cell biology and cellular organization, division, and processes. And we already had one problem solving session in the previous week where we discussed the initial lectures for this course. Now, in today's class, we will do the lectures for the second week, which was these. So in the first week, we looked into DNA, we looked into cell. So what we'll do in this week, if you have seen the lecture note, it basically is about the regulation of the cell cycle and the checkpoints. So we'll have a look at the cell cycle. How does the cell cycle look? What are the different stages? And then we will see the checkpoints and the regulation of this cell cycle. As we know that this is a regulated mechanism. It is under regulation and is under checkpoint control. So we'll look into all of these details. As you will remember, the 4 lectures that were for this week were regulation of the cell cycle, and then about the checkpoints, which talks about DNA damage and DNA replication checkpoints. And then the system which is also responsible for degradation of the cyclins and the CDKs, if you remember, the ubiquitin proteasome system. So we'll also do that. Okay. Now, like last time, I will not be going over into the details of each of these topics in much greater depth. That's because the session is more focused on giving you question answers or questions which can be used for your map for your exams. But if you have any particular doubt or any topic that was not clear to you, do let me know. I can take that up as a topic to discuss, right? So let me begin with today's session. Okay, so just a quick refresher of what is cell cycle that we already know of. So the cell cycle was discovered by Prevost and Dumas, and this was discovered by studying the cleavage of zygote of frogs. So if they had looked at the cell divisions of the zygote, and they realized that how the cell divides from one stage to another, what is the timeline, how does it take place, as you can see here. So these are the cleavage. These give rise to different stages that we already know of. We have heard these terms multiple times. We know there is an X morula, blastula, gastrula, if you remember. So all of these are different stages, and while looking at these different cell stages is when they realized that there was a highly regulated mechanism that takes control of all these different processes. And that's when they came up with the theory of cell cycle, which is basically a series of stages that a cell passes through in order to divide and produce new cells, right? And if you now look into the cell cycle, this is how the cell cycle looks. This is what has been discussed in the lecture videos also. You will see that, so this is a lifeline of a cell. You have the cell in its entirety is divided into cell growth, DNA synthesis, and cell growth again. And then the period between this phase and this phase is called the mitotic phase. Okay, so let's begin. We will have first as the G1 phase. G1 phase is basically the growth 1 phase. So this is cell growth phase, followed by something called DNA synthesis phase, which is the S phase. And then we again have growth phase, which is G2 phase. Then it is followed by mitotic phase, where the cell divides into two new cells and or divides into different cells and gives away the replicated DNA, which is synthesized here. So here, let's say we all are diploid organisms, so we all have 2n DNA content. So here you will have 4n DNA content, and that those 4n content will be then passed on as 2n and 2n to the new daughter cells. And that happens at the mitotic phase, which consists of mitosis, which is the process of division of division of cells and the passing on of nuclear material, followed by cytokinesis that we will see. right? So this except for the mitotic phase, the entire other stages, which is G2, S, and G1, are together called the interphase. So you can divide the cell cycle as two steps. You have G phase, sorry, you have interphase, and just one second. I have to few. Yes. So you have interphase and mitotic phase, and then in the interphase you have G1, S, and G2. Okay, so this is the basic that you should remember. Okay, and why do you at all need this cell cycle? So I told you about the regulation of cell cycle. And we know that this is required. The cell cycle was discovered as a during the cell division process. But why do you need this cell division? Why do you need this cell cycle at all? So we need that the cells needed for the development and growth. So you have a single cell, which is what every organism begins with as a single zygote, and you form that multicellular organism after for several rounds of cell division. So you need cell cycle for controlled development and growth. Then you need them for cell replacement. So your cell division actually replaces old cells with new one. So you also need the older cells to die off and the newer cells to take their place. So you need again cell division there. You also need regeneration, which is in the case of Hydra. If you know that if you basically if you divide the body into two parts, each part can regrow into a whole organism. So that's also because of cell division. And lastly, you have asexual reproduction, where organism cells, they divide to form new cells, and that those new cells give rise to new organisms. So you have, so basically this just highlights the importance of cell cycle in the span of the cells, right? So that's why, because of these important roles and because of this very important contribution to the life cycle of a cell, this cell cycle mechanism is under high regulation and is a very controlled mechanism. And we will see why and how do this does that take place. Right. Now, let me just quickly take you over the different phases, right? So you have gap 1 or growth 1, S1, and growth 2 or gap 2 phase. And then you have the M phase. So if you have to just quickly give some names or quickly associate some words with these stages so that you can remember quickly, you can. So always try to remember it from G1 so that it takes you to the entire cycle. So G1 is basically growth and metabolic, normal metabolic roles. Then you have the DNA replication, doubling of the DNA content. So S phase is basically synthesis phase. Then you again have gap 2, G2 or growth phase 2, which is basically growth again. So the cell grows in size and then it prepares for the upcoming stage, which is mitosis. In mitosis, you have 4 different stages. You have prophase, metaphase, anaphase, and telophase. So we'll see this in all these different stages just for a quick refresher. So let's start with G1 phases. So during G1 phase, which is growth 1 phase, the cell grows physically larger, which makes sense, copies organelles and makes the molecular building blocks it will need in later steps. So when your cell is dividing, what is the ultimate idea? That the content has to be divided equally into the new daughter cells. So whatever is in the cell needs to start making copy of itself, needs to grow so that you can have this division taking place. So the growth phases do that. So in the growth phase, the cell grows and makes copy of its organelle and everything else that it requires to pass it on to the next generation. Then comes the S phase, which is basically the DNA replication phase, where you have the whole process of DNA replication that you would have learned in your biochemistry or in your structural biology. How does the DNA replication take place? You have the polymerase coming in and you have the primase and all that stuff that takes place in this stage here, S phase. So you have your DNA replication, which basically doubles the quantity of DNA in your cell, makes it from 2N to 4N, right? And along with DNA, your centriole also divides into two centriole pairs. We'll see why, because you need those at the two other cells to pull the DNA into the new cell. We'll see that in the mitotic phase, right? So everything associated with DNA gets replicated in this phase. And then again, you have growth phase 2, which is basically during this phase, again, the cell will grow, make more proteins, begin to reorganize its contents for mitosis. So these growth phases are basically making the cells amenable for the next stages. Here it makes fit for the GS phase, and here it makes more for the mitotic phase. Okay, now there is another phase which you remember professor talking about, which is called G naught phase. So G naught phase is here. Okay, so G1S, G2, there is a phase here called G naught, which is called cell cycle arrest. Okay, so when you have G1 phase, after that, there are some quiescent, senescent, and non-proliferative multicellular eukaryotic cell which never divide or don't divide until a signal comes. So they are arrested in their cell cycle phase, and those are G naught phase. So if there is a cell which is arrested in its cell cycle, it is arrested at this phase. Okay, so DNA content remains the same. The cell has just divided after mitosis. It has formed a new cell, and there are cellular components that have reorganized, and it stops at that phase, right? So it's basically in those cells that don't need to don't need to replicate that many times. They're fully differentiated cells. Okay, if there are cells that require continuous replenishment, they never enter G naught, because obviously to break from G naught also they require several cues. Some of them don't ever come out. But if you have a replenishing cell, you would never enter G naught phase, and you don't have to be under any condition. You simply follow through G1S and G2. They're timed, of course, but the ones that enter G naught, for example, neuron, they remain in that phase for either long period or refrain indefinitely until any cue comes, right? So G naught is basically when the cell stops dividing and gets arrested, right? Okay. Now coming quickly to mitotic phase for a quick refresher of all the different phases that are there. You have prophase, you have metaphase, you have anaphase and telophase. So in case someone is not very well aware, I will just quickly walk you through. So basically here in this mitotic phase, mitosis takes place. Mitosis is division of cell, equal division of cells. You have equally cell dividing into two parts. So you have 4N already, which is formed in the S phase. The 4N DNA content is now divided equally into the daughter cell as 2N and 2N. That's what you want. Okay. So what happens in the prophase, which is the first phase, your chromosomes start to condense. So basically, if you can see here, your chromosomes have become thicker. Chromosomes, why? Because there are 23 pairs in ours, right? We have 46 chromosomes. So all these chromosomes, all this DNA have started thickening, and they are now forming a plate. Okay, we'll see that in the metaphase in a while. Then you have this mitotic spindle which is formed, and this spindle grows between the centrosomes as they apart. So these are centrosomes. Remember I told about centrioles, centrosomes there. So these are centrosomes, and these are connected by these mitotic spindles. And what you see is these two centrosomes are moving apart. One is going to this pole, one is going towards this pole, and these mitotic spindles are in the process getting enlarged. Okay, and the nuclear membrane and the nucleus as an organelle starts getting disrupted. So you have the nuclear wall breakdown, the nucleolus getting disappeared. You have this every organ, because you want to facilitate the DNA moving to two different poles, right? So nucleus has to get disintegrated, right? So that's the beginning. Okay, everything is forming and it's moving towards the other side. Then you have pro-metaphase before the metaphase, right? Here, the chromosomes that were forming, that were condensing here becomes even more condensed, and they become this compact material. So as you can see here, they have, so you would have seen in your cell cycle or in your metaphase some structures like this. Those are formed here, as you can see. So the long DNA has started taking this V-like shape. Okay. And like I said, the nuclear envelope has broken down, and the mitotic spindle, as you can see, they have grown, and they have now reached here on the opposite side, and they have started capturing the chromosome and kinetochore on the centromere of each sister chromatid. What is sister chromatid? It's basically the copy. Okay, so each of the cells, each of the chromosomes have a sister chromatid, right? So the two poles have kinetochores. Sorry, the two poles have centrosomes, which have mitotic spindles, and the chromosomes have, chromatid is one chromosome. Like chromosome, when we say both of them together. When we just want to talk about one unit, we talk chromatid. So when we talk about one sister chromatid, they have kinetochore. So those spindle come and attach on the kinetochore. So you can imagine it's like a pulley at both sides, and they have all connected to their sockets. Okay. Right. Next, what happens in metaphases? So these are all at different places in the like cell. So what they do is they align in this central part of the cell, which is called the metaphase plate or the equatorial plane. And you have the two kinetochores of the two sisters, 2Y, because we are 2N. We have two copies of each chromosome pair. So we have, so those two kinetochores are of each chromosomes are attached to via microtubules to the opposite spindle fibers. Okay. Now we have anaphase. In anaphase, what you see? You see the pulling. So the sister chromatids separate from each other and are pulled towards the opposite side. And the protein holding the sister chromatids together is broken down, allowing them to separate each now its own chromosome. So whatever was here has now broken into two parts and they have moved. Each chromosome is moving towards the other side, right? And remember, they're already double, so they will go there and they will form, because they're already double, they will be actually so the 2n is retained here. This here was 4n. They have now become 2n again, right? And the sister chromatids have separate. That makes sense because then you have equal distribution, right? So your sister chromatids have moved apart. The attachment between the sister chromatids is broken and they have grown that side. And you have telophase, which is basically reorganizing of cell, and cytokinesis is basically the furrow which breaks down these two cells. Okay, so telophase is basically in their so they move to their poles. They start forming two new cells again, and cytokinesis, and when the two new cells break apart, right? So that completes your mitotic and meiosis phase. Now, let's try to understand these with even more technical terms, which are questions taken from assignments of this term, previous term, and previous question paper. So let's try to answer them so that we get a good hold of these topics. Right. So the cell cycle is best. So we have, for today's session, I have numbered these questions so that we have ease of discussions. So maybe we can start attempting these questions. So you can take your give your options in the answer box, chat box, and we can discuss the questions if you have any doubt. These are fairly set of easy questions. We'll later be coming into cyclenes and CDKs where you need to remember particular pairs of the combination. So these are fairly easy questions. You can take the chat box for your answers. We'll wait for a few seconds and then we can move ahead. Okay, so once the sequence of events, okay, next. Anyone else wants to take a try? Yeah. F1, C, D. Okay. 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 So I do have responses. I'll wait for 1 more minute. Maybe people are still typing. All right. So I see a lot of responses. Maybe I'll just go to the answers. Right? So many people said C, D, C, B, which is correct. Let me just check the answers quickly. Yes, like I said, there are some different answers, but we can just go over that quickly, right? So the cell cycle is best defined as sequence of events from one cell division to a next, which people gets right. Next is longest phase is interphase. Interphase, as you remember, consists of all the other stages except for M phases, and G1 is primarily associated with protein synthesis and cell growth. Rest everything is associated with DNA, chromosome, DNA replication, spindle. So this is S phase, this is segregation, this is I guess in S phase. This is actually in mitotic phase, spindle formation, which also come later. So it's mostly the growth phase is mostly associated with protein synthesis and cell growth. Cell growth is the key. Okay. Okay, let's move on. So cells enters G naught phase when? When do they enter G naught phase? Which step has DNA replication? And then which cell? So if the which cells actually enter G naught cells and they enter permanently. So they do not have to return or they don't have to go back to growth phase. And then which stage has doubling of centrosomes? These are also pretty simple questions because these are direct question based on the stages or the different phases of cell cycle. CBCB. Okay. Um, CBCB. S is B basically, right? C, I get a lot of C. B, yeah. Anyone else? That's it. Okay. So the answers are this, which again, most of you got right. So cells enter G naught phase when they exit the cell cycle. This is what I told you. When they are done with their M phase and they're supposed to enter G1 again, they will enter into G naught if they don't want to undergo the cell cycle replication again. Right? DNA replication, this is very easy, S phase. Doubling of centrosomes also I told you is an S phase. And which cell commonly remain in G naught cell permanently? It is neurons. So people have answered neuron. That's the correct answer. And like I said, terminally differentiated cells. Terminally differentiated cells means they have reached the end step, right? They don't have any other uh, cell to be what is differentiated cell or what is undifferentiated cell. When you are for example morula, followed by morula you have blastula, blastula followed by that you have gastrula. So you in neurons we don't have to do worry about that. That's the terminally differentiated. That's the most differentiated cell in that cell line commitment can have. So they are differentiated cells and that's why they exit the cell cycle into G naught permanently, right? Talking about the other side cells that I have just mentioned, for example, hepatocytes, they are usually in G naught, but they can re-enter the cell cycle if there is liver regeneration, right? We know that liver can regenerate it. So if there is liver regeneration, then these hepatocytes, which are otherwise in G naught, can re-enter cell cycle. Uh, Next one we in the options which you have is fibroblasts. So this fibroblasts, they are quiescent, they are also G naught, but they can also proliferate, and that comes in form of stimulation. So fibroblasts you will generally find associated with wound healing. So if you have any wound healing procedure that is to be done, so these cells can re-enter cell cycle. Okay, so the next one was stem cycle that we know that these are quiescent, they don't replicate, but they do have proliferative capacity, and we know that, right? So the question was something which is permanently in G naught, and that is neurons. Okay, all right. Let's move ahead. Okay, next set of questions, which is basically G naught, sorry, G2. So what does the G2 prepare the cell for? What is the correct sequence of mitosis? Okay, and then correct sequence of mitotic cell cycle, and then chromosome condensation begins in which phase of mitotic stage, right? Easy questions, but it gives you a more grasp over the topic. If you have question, I might not be looking in the chat box. I mean, I might not be looking at the screen. If you have any doubt, put it in the chat box. I will definitely come and read here. But you can unmute and talk anytime you want. I might not see raised hands, so that's something that I just wanted to tell. You can use the chat box for any problems or doubts that you have. Someone's raising hand. Do you have any question? Eva, do you have any question or do you want to write something? Okay. Yeah. What is apoptosis? Cell death. Do you have any specific question related to that apoptosis? I mean, just to define the term, apoptosis is a programmed cell death where a cell which is of under stress or which is destined to undergo death, then there's a programmed cell death, which is basically all these factors which disrupt the cell membrane and then kill the cell organelles. Those come into play. So apoptosis is a highly regulated and highly controlled cell death mechanism. If you have any particular question with particular to any topic, then please write it down. Otherwise, I hope that suffices the question. Please let me know if you have any other doubt. If you have any other doubt, then please let me know. Okay. All right. So let me give you the answers. We have stopped at this slide for far too long. Okay. So I think most of you got it right. G2 phase prepares it for the upcoming phase, which is mitosis phase. Chromosome condensation, we have already discussed, is in the first phase, which is prophase. Correct sequence is pro, meta, and telophase. And then for cell cycle, it is G1S, G2M, which most of you got right. So that was great. Okay, the last few questions for this, which only talks about the cell cycle stages, to get more clarity on these topics, right? Sister chromatids separate during which phase? Nuclear envelope reforms in which stage? Chromosomes align at the equatorial plate during which of these stages? And the fusion of S phase and G1 cells induces what? Now, this question gives you a uh, how do I say, an idea of what I'm going to discuss next. Caspases, right? If you just hold on with me for some time, then when we will be discussing the ubiquitin process, ubiquitin proteasome, I will discuss apoptosis and caspases there. We have this discussion coming up at the end because that's anyway going to be discussed about the ubiquitin system. So I will talk about apoptosis and caspases there and what does it refer when, how are they related to cell deaths. I hope that's okay. CBDC, CB, okay. CBDC, right. Okay. Uh, I've received most of these answers, right? So let me just. Right. So most of you have written CBDC, which is of course CBDC is correct. So you have sister chromatids which are separating during the anaphase. Chromosomes will align at the equatorial plane in metaphase. These are correct. Nuclear envelope reforms in the telophase, that is the ending step just before cytokinesis. Now this, most of you have gotten right, which is great. So you have fuse a S phase and a G1 cell, which induces DNA replication in G1 nucleus, right? And this tells us about something which is related to the cell cycle control, right? And we'll see how. But before that, just a quick overview. Like I said, cell cycle is a highly controlled and regulated process. So there is several steps in the cell cycle which is tightly controlled by checkpoint. Checkpoints means at those process or at those steps, uh, the decision if the cell cycle will continue into the next phase will take place, right? So let's say you are in the junction of M phase and G2 phase. So should your cell commit to the M phase, is taken, that call is taken by the checkpoints, right? So that's the kind of mechanistic control that we have. in cell cycle, right? So these cell cycle checkpoints, they ensure, what do they ensure? They ensure that there is completion of the different steps in the previous stages. And if there is any DNA damage that has occurred in the cell, that repair has to be taken place, right? So like I said, one of the checkpoints is G2 to M, which is commitment to the M phase. The one of the others are G1 to S, which is commitment to the S phase. And then you have one in the mitotic phase where your cell is undergoing division, right? And then each of these checkpoints is controlled by the mutual interaction between a cyclin and a cyclin dependent protein kinase. So we have a cyclin and we have a dependent kinase, which is called a CDK, which together make up this complex that you all know, but I will not name it now because we will be discussing the experiment in the next slides. So that complex is what determines if the cell should go into the next stage or should be staying here at the stage and correcting the problem that is there, right? And P53, which is a protein that you will keep coming across, P53 gene products are known to control many events through these checkpoints. So you have this complex that we know of already, but we have other proteins that assist these complex in their checkpoint activity, right? So you can see here you have, like I said, which will do in the coming slide. You need to remember these pairs, okay? Which cyclin and which CDK goes together, and then which proteins are the ultimate associate or accessory proteins that help in the process of cell cycle control, right? But before I do that, there were few experiments that were mentioned by professor in the lecture, right? So we will do that quickly so that you understand how did this discovery happen, because these experiments are important in the perspective of the week 2 lectures, right? So let me just begin with this. Do you recognize this experiment, which was conducted by Johnson and Rao? So what was their observation? So which key observation demonstrated that diffusible factors in the cytoplasm regulate the progression of the cell cycle? I will wait for your answers, and then of course in the next slide we will discuss the experiment also. Any response? Anyone wants to take a guess? The experiment was fairly simple to understand, but gave an observation that was key to the determination of how cell cycle functions. Anyone wants to attempt? So let me read out the option. Maybe reading out the option can just tell you what's happening. So try to remember the experiment if you are not able to recollect. So it says that out of the options, G1 nuclei immediately began DNA synthesis when fused with cells in the S phase, indicated S phase promoting factors in the cytoplasm. G2 nuclei began DNA synthesis when fused with S cells. So was it G1 fusing with G2 or G2 phasing with G1 fusing with S or G2 phasing with S that gave the observation? Third one is G1 and G2 nuclei failed to influence each other, proving that the cell cycle are independent. And last one is DNA synthesis stopped in the S phase upon fusion in mitotic cells. To try to remember the combination that was done and then try to remember what did they say, and then that basically takes you to the observation and the name that was given to the complex. Yeah, I have responses. I have A, right? Okay. I will just move ahead. Like people have mentioned, it's A, which is basically G1 nuclei immediately began DNA synthesis when they are fused with S phase, right? So let me talk about the experiment once again. So this was done by Johnson and Rao, and this led to the discovery of MPF. We will know what is MPF in a while. But before that, let me just tell you what the experiment was all about, right? So this experiment, the goal of the experiment here was to see if cell cycle progression is controlled by any factor, particularly cytoplasmic factors. So the technique used here was cell fusion, like it's already mentioned in the options, right? That they use cell fusion. And what they did is they used mammalian cells at different cell cycles. So they had cell cycle states. So G1, S, G2, S, M, all this. And then after fusion, if you're fusing two cells, they are sharing a common cytoplasm. And that's the key here, that if anything which controls the cell cycle is present in the cytoplasm, that should be able to influence the next phase. Okay. So the three fusions that actually took place there was S and G1. Okay, S and G1, S and G2, and then M phase cell with all of these three differentiated cells. So now just try to imagine the cell cycle that I showed in the first slide, where you have G1, S, G2, and M. Now what you did is keep the M and fuse it with every cell. That's done. Then take the S, fuse it once with G1 and once with G2, and we'll see what happens. Okay. So when your S phase was fused with G1 cell, so G1, which is otherwise before S phase. So when you are in G1, when you complete your G1, then only you enter in S phase, right? But when you have a S phase and a G1 cell, and you fuse them, the G1 cell immediately starts DNA replication, which is something which is occurring before a particular time, right? So what is the inference from this? That the S phase cytoplasm has a replication promoting factor because of which the G1 started replicating. Even though it is supposed to wait and grow and then complete its cell cycle, it just quickly started replicating. So this is something called replication promoting factor. Okay. Now this happened to G1. So let's try with G2, which is on the next step after S phase. So you fuse G2 and S, but what you should expect, the G2 should also start replicating, and that did not happen. What did that confirm? That G2 already is in a phase which is already with replicated DNA. So once your replication has happened, DNA will not go into re-replication. What does it ensure? It ensures that the cell automatically does not go from a 4N condition to an 8N condition. That's why your cell remains at 2N, goes to 4N and immediately goes to 2N 2N condition. It will never be from 4N to 8N or 2N to 8N, nothing like that, because re-replication is stopped. And that's because of this observation that your S phase and your G2 does not go in parallel with what happens when S and G1 are fused. Right. And then you have the last set of observation, which is from the M phase cell and its fusion with the other stages. What you see is the interphase cell, which is all this S, G2 and G1, they enter mitosis. So you have a promoting factor in S cytoplasm and in M cytoplasm, right? So what happens when you have your G1, G2 with M? You have immediate chromosome condensation, nuclear envelope breakdown, which is exactly what happens when the cell enters mitosis phase. So your M phase cytoplasm also has a mitosis inducing factor. So there is a replication promoting factor and there is a mitosis inducing factor, which they named as maturation promoting factor or mitosis promoting factor, and that's called MPF. I hope that is you are able to recollect that the professor also told about this, that the complex that regulates the cell cycle mainly is called the MPF. Okay, right. So they knew about the presence of this factor. They knew about the major role that these MPF plays, which is basically that they can it's a dominant activity, so it can force any nucleus to undergo mitosis. But cell cycle control is very phase specific and factor dependent, because not every phase will undergo replication. Once a previous step is completed, it will not go back to its previous step, right? So overall, why is this experiment important? Because first, it is a proof that there is a mitosis inducing cytoplasmic factor. And then that's why the term discovery of MPF, because MPF was discovered by them. And later, further experiment found out that these MPF consist of cyclins and CDK, right? But the basis of this foundation, or the foundation of this complex existence, came from this experiment, right? Which is called Johnson's and Rao experiment. Okay, so one experiment that you should remember. This is the other experiment that was done. So before I explain the experiment, do you remember anything from that experiment particularly? So this is the Masui and the Marquardt experiment with frog oocytes. And what was the observation? What was observed when the cytoplasm from the M phase cell was injected into immature site? Now we know there is an MPF. We wanted to understand the MPF better. So what these scientists did, they took the MPF, or the M phase cytoplasm extract, and they inserted into immature oocyte. What was the observation that they found? B? Anyone else? A. It is A. Interesting. So I have B and A. Anyone else? B. Okay. Mm. Okay, so I have different answers. Let me give you the answers first. So the correct answer is the immature oocyte got matured and entered M phase. Now people have also responded by saying that it stopped growing and it entered DNA replication. And let me tell you why that was wrong, and then I will explain the experiment. Okay. So the experiment here, which is basically Maswin and Marquardt, they basically took the cytoplasmic extract from M phase, okay, and inserted it into an immature oocyte. The immature oocyte, if you remember from the lecture note, these were arrested, immature, because they were arrested in prophase 1 of meiosis. So they were already in the prophase stage of meiosis when the M phase cytoplasm was entered or were injected into these frogs, oocytes. These activated the active, so basically these activated the mitosis phase, like M phase was activated because of the presence of MPF, right? And what happened is the oocyte then underwent different vesicle breakdown and then entered into M phase, meiotic maturation, right? So why did not it take DNA replication like people mentioned? It basically means that if it underwent DNA replication, which is presence of S phase factors, but that is coming from the S phase. We, the scientists here, are injected M phase. M phase has factors that promote M phase, which is mitosis followed by meiosis. So if you have to do DNA replication, you need to take S phase extracts, S phase stage extract, right? And then it also says someone said stop growing, which is not correct because they did observe maturation and they did observe meiosis, and that confirmed the theory or the hypothesis proposed by Johnson and Rao. There are some factors in the M cells that promote. The idea here is that you have factors that promote the steps. So you have a certain factor that promotes mitosis. You have a certain factor that promotes the replication, right? And there was another option which said remain in G1 phase, which is also not correct as we now know of, right? Okay, now let me just walk you through to the experiment. So this experiment, as you know, before this experiment, as you can see, there is a timeline gap here. So in 1965, around 1965, we had the Johnson and Rao experiment done. We had the conclusion. We knew that there were some factors in the M phase and the S phase which could able to uh, promote replication and maturation in the maturation, or for that matter, mitosis in the cells that they are injected in, right? So what the Masui and Marker do in 1971, they basically wanted to determine whether mature oocytes contain a cytoplasmic factor that trigger entry into M phase. Okay, this is not from like S phase or G2 phase or something. We are taking mature cells. Okay, now, as I told you, they took immature cells because you're comparing a mature cell and want to see if your immature cell behaves like a mature cell if I take extract from mature cells, right? So the cells that they were testing on was immature xenopus oocytes. They were arrested in the prophase 1 of meiosis. So it's not just if you we discussed only mitosis. There is also meiosis that happens in the M phase where you enter into meiosis 1, meiosis 2. They also have their own prophase, anaphase, telophase, and anaphase and telophase and metaphase. So these cells, the immature cells, were arrested in the prophase 1 of meiosis, which is meiosis stage 1. Okay. So uh, what happened in these kind of cells, which are already in these stages, they require hormonal stimulation, for example, progesterone, and these cells which are arrested in the meiosis, this is similar to in humans also, where you have the X cells which are arrested in a certain stage of cell cycle, and when they are stimulated, that's when the replication or the whole cell division starts, right? Similar concept here as well. You have this immature oocyte that require hormonal stimulation, and then it undergoes oocyte maturation and everything else, and so that they can form the meiosis that leads to further fertilization. Okay. Now, basically, you need a hormone to overdrive the arrest. So what they wanted to check, if the extract from the mature phase, which has already undergone everything here, does that have a factor that can override the hormonal requirement? Can we have the same oocyte maturation without hormone, but the presence of just a mature cell extract? That's what they did. They took cytoplasm from a mature phase, oocytes, and then they injected it into the immature oocyte, and the response was seen as entry of these immature cells into M phase. Okay, and then if you do it repeatedly in serial injection, you will still see the same factor. Right. Now, what did it say? It said that the mature oocyte cytoplasm, it also has that MPF, which was seen in the Johnson and Rao experiment. And hence, the MPF is now experimentally proven that it can facilitate mitosis. Right? It was a hypothesis, or it was an observation based on different cell cycles or cell stages. Here you have two different cell stages, but you have now, instead of fusion, you have taken extract and directly entered in the cell. So if you are fusing, you can also say that the factor was already present in the other cell, and that's how it got activated. So there was no fusion experiment. It was direct injection experiment. You take extract from the other cell and just inject and see if you can see the same factor. If the factor is present in the cytoplasm, it should give the same effect, and it did. And that's how it confirmed the presence of MPF in the cytoplasm. That's what Masui and markers did. They confirmed the presence of MPF in the cytoplasmic factors, right? Okay. So let's do some questions to understand this further. Okay, so the Johnson and Rao experiment. So you have Johnson and Rao, what did they show? And then you have the Masui and Markert, what did they show MPF was? And then you have a question which says, when cytoplasm from M phase oocyte is micro-injected into G2 phase oocyte, what could be the possible effect? Which is very easy now if you just recollect the steps that we just discussed. There are many options that look similar. A key for you people when you are doing exam question. We know that it promotes mitosis. We know that it promotes replication. There will be many answers which are in and around those options. Some will break away it into saying different stages of mitosis. Stick to the original observation. Whatever words was used in the original observation, just use those as your reference and answer that question. C. C is for which answer? There are three questions in this on the slide. Mm. I see people writing C, but there are three questions on this slide, right? So all of them are C. Just write all C, something like that. Yeah, C, C, C. Everyone's writing C. That's great. Right. That just shows that everyone has understood it. Great. Right. So I don't need to explain it further as you would have already understood. Johnson and Rao experiment demonstrated replication promoting factor in S phase, not MPF activity that came from Masui and Marker's experiment. The activity part came from the other experiment, right? Masui and Marker showed that MPF is a cytoplasmic regulator of mitosis because they took the cytoplasmic extract from the mature oocyte. Okay, next one. When you have a cytoplasm from M phase oocyte into G2, it will start behavior or it will be driven to M phase, which is great. Everyone got tried. Next few questions, and then we move on to the next topic. So MPF, now that we know about MPF, before I jump to the next topic, let's try to answer these questions. So MPF is composed of which component? MPF activity is peak during which of the cell cycle? And injection of cytoplasm from an M phase, I think this is already done. But yeah, you can just try to attempt it once again. You will see that these questions are fairly similar or repetitive. That's because I tried to collect it from different years of assignments and exam papers. So that's assignments and exam papers. So basically, that's why there are some repetitions. But yeah, you will see the wordings are different or some minute differences are there, which just helps you with understanding the type of questions that can come. Mm, yes. CDC. Okay. Anyone else? CDC, right? Most of you have written that. Okay. I just wait for 1 minute more if anyone else has to answer. Okay. I will go ahead. Right. CDC, as people have mentioned. So MPF is composed of CDK plus cytidine that we already know of. So basically, coming to the next question, MPF activity is peaking during the M phase, and we will see that uh, during your S and G2, there is cyclin B accumulation. MPF becomes fully active in the G2 to M transition. Okay, and your MPF is basically cyclin B and CDK1 together. Okay. And then MPF, coming back to MPF activity, which is highest during mitosis, that's where it causes the chromosome condensation and nuclear envelope breakdown and everything that is to be done with your mitosis happens at the peak during M phase. So that's why it is the cycle, the concentration very different. The activity of the MPF is highest in the M phase, right? And the third one is basically the same thing that we have already spoken about, that there is an active MPF in M phase, which is when injected into the G2 phase, causes its entry into mitosis phase, right? Okay. So now let's come to the MPFs that we know of, cyclins and then cyclin dependent kinases. Okay. Let's just first try to understand what is happening here, right? So uh, basically, like I told you, that there are these complexes that promote the entry into the next stage, right? So let's start. Let's say you have G1. So this is I told you, right, to commit to the next point. So you have a cell which is in G phase and has to enter S phase. The decision to enter, whether to enter S phase or not, happens at this point here, which is somewhere past the G1 phase. Okay, so you have the towards the end of G1, you have this checkpoint, and then you take the commitment of going into S1 phase or not. Now, here during G1, what drives the G1 phase? You have cyclin D and CDK4 or 6. Remember these combinations are very important. Which cyclin goes with which CDK and in which phase? This is what you should remember. Okay. So you have cyclin D, which goes with CDK4 and 6 in G1. Then you have a cyclin E and CDK2 complex, which takes the cell from G1 to S phase, right? That's the checkpoint of G1 and S. So what happens there is your cyclin D, as you can see here. So let's try to see here. These we'll do it together so that we understand the concentration of the cyclin. The first thing is cyclin. The name cyclin is because of the fact that it undergoes a cyclic concentration changes, right? That's why cyclin. Okay, and you can see from the image here, it's cyclic concentration changes, right? So let's do cyclin D. Okay, it begins synthesizing at cyclin D, and it stays and then reduces at the M stage. So all these while cyclin D is going higher in concentration to a lower concentration, right? Cyclin D's cyclicity is maintained in that way. Cyclin E comes up at the end of G1 phase, which is here, and then goes down at the beginning of GS phase, which is basically mean that this is trying to facilitate the entry into S phase. Okay, so D and then E. Okay, the next phase, which is which drives the S phase, is cyclin A. So you will see the cyclin A concentration increase in this S phase remains high towards for the whole G2 phase and then goes towards the end or beginning of mitosis. So these two phases are driven by cyclin A. Okay, what changes? changes happens in the partner. First, it is binding with CDK2. In this G2, it binds with CDK1. Okay. And where does it take CDK2 from? It takes CDK2 from CDE. So what I want, or what I wanted to show here is that you should imagine how this like interaction between CDK and cyclin is taking place in a regulated manner to drive the cell cycle. If CDK2 is binding with cyclin E, it takes it into S phase. Cyclin E go down, it binds to cyclin A. It takes to G1 and S2. Then this CDK goes down. CDK1 comes and it goes to the G phase. So it is just a cyclic interaction between these components that drives the cell cycle. And that's how it is very well regulated, right? Okay. Now coming again to this. Right, so we are here, right? Now there is another checkpoint here which makes sure that the cell has duplicated, the DNA has duplicated, the cell has grown in size and everything is taking place. And it is okay for the cell to now undergo DNA division and the cell division, right? So here the checkpoints are cyclin A and CDK1. And at the same time, you have an increase in cyclin B. And that cyclin B now drives it to the M phase, right? So you have D, E, A, B. Okay. And you need to remember the partners, cyclin D, CDK4, cyclin E, CDK2, cyclin A, CDK2, cyclin A, CDK1 towards the end, and then cyclin B, CDK1. Okay. Then you also have a checkpoint where you enter from the mitosis phase, right? So what does this say? That they are under the cyclic regulation and it's a very highly controlled mechanism. Again, you will see the same conditions here. Now, if I have to just explain it to you in terms which are amenable for, like the technical terms that I should use, let me just tell you what does CDK do? Why is it partnering up with CDK in different cases, right? CDKs are basically cyclin-dependent kinases. What are kinases? They are protein kinases, okay? What do they do? These CDKs, they are present at constant level throughout the cell cycle. As you can see, the name cyclin is for the component, which is the cyclin A, cyclin B, and they are in cyclic concentration cycle. That's why they are named cyclin. The others have a constant concentration throughout the cell cycle. However, they are inactive on their own. So CDKs cannot do anything on their own. CDKs need to bind to the cyclins so that they can do their activity. What is kinase? We'll see in a while. Okay. And the cyclin, like I told, their level rise and fall. So their concentration is what determines which phase your cells will be in, and they bind to the CDK to activate them. Right. Now, what happens? When cyclin binds to CDK, as you can see on the slide also, there is a partial activation of this CDK, right? Now, there is what happens following this is there is something called activating phosphorylation. You have a phosphoryl group which comes which basically to the threonine 161, you have phosphorylation of that site by CAKs. We will see cyclin activating kinase or CDK activating kinase. Kinase is anything that attaches phosphoryl groups or phosphate groups, right? Okay. Now, for the CDK, binding of cyclin and the coming of CAKs to phosphorylate the threonine 161 together makes it an activated compound, right? Along with this, there are some inhibitory phosphatases which are removed. So, for example, you have tyrosine 15, threonine 14. These phosphates are removed by CDC25. So, there are some phosphates that are blocking. So, you remove that. There are needs for some phosphate that is activating it. So, it activates it. And then together, it makes up the fully active CDK cyclin complex. Okay. So, now you need to remember these CDKs. In the middle, I also told you CDC25. These are concomitant. They go hand in hand. The names are CDC and CAKs and CDKs. They come from the system that they were discovered in. Okay. Most we will see CDCs are discovered in yeast sign, and yeast was discussed majorly in the lecture notes. So please do pay attention to which CDCs refer to which CDKs. Okay. Okay. So there are a few more terms that I would like to mention here because I think the next slide talks about them in questions. Just a second, let me check. Yes, we'll do, of course, question, and with each question, it becomes more clearer. But just to give you a heads up of what's exactly happening. So in general, when you have a regulation of this CDK cyclin complex, what does happen? You have cyclin synthesis as we just saw, and there is synthesis of cyclin and degradation of cyclin. Okay, that degradation, that constant fall that we kept seeing, that is because of ubiquitin proteasome system. That is the last thing that we will discuss today. Okay, and so proteasome, that's why you see the importance of ubiquitin and proteasome. If they don't act, they don't take the cyclin down. So if I just take you back to that slide here, if they don't take cyclin E down, cyclin A will not come up. And if cyclin E is higher, this will not function. So if cyclin E doesn't go down, the cell doesn't enter S phase. So cyclin E needs to be degraded, and that's why you need ubiquitin system also in play. So you see all of these systems are complementing each other, and they all needs to ensure, they all needs to work in their correct way, right? So you have cyclin control. Next, I said phosphorylation control. So there are some names that you would have come across in the lecture notes. For example, V1, CDC25, phosphatase, and CAKs. One key, one just like one key for you to try to remember what the enzyme or the protein does. Generally, in the history of naming of proteins, the name of the protein is given to the phenotype it is, which is with what is observed, right? So V1, basically, if you mutate the particular protein, then the cell don't reproduce, cell don't grow, this remains small. So if you mutate that protein, it becomes small. So you have named that protein V1. Okay, so you can remember it like this. Most of the times, your proteins that are named, especially in developmental biology, comes from the mutant phenotype. So you remove the activity of the gene, and then you see the phenotype of whatever you're observing. And based on that observation, you name the cell. So V1 basically tells you that that gene is actually responsible for growth of the cell. That's how you can remember if you ever come across some confusion terms like this, right? Most of the time, they will look very counterintuitive. Why are you talking about V when the cell growth, it is responsible for cell growth? But the name of the protein comes from the mutant phenotype. Okay, so that's how you have V1 kinase, which is basically removal of the inhibitory phosphorylation. CDC25 also removes inhibitory phosphatase, and then CAKs, which activates phosphorylation. So CAKs is, as you know, CDK activating kinase. CDC phosphatase removal, so it removes the phosphatases. So all of these taking place, and then you also have uh, I don't know how many of you have read biochemistry, but these are all very related concepts. So if you understand one, the other becomes very easy to imagine. So if you have seen biochemistry, and if you have studied glycolysis, or if you have studied TCA or any all these ETCs, which we also touched upon in the last session, you know that every step, so if there is breakdown of glycogen tends to be taking place, it is under hormonal control. It is that hormonal control is itself maintained by the concentration of the end products. So what does happen when your glycogen breaks down? You have glucose. That means your body is less in glucose. That's why you need glycogen to break up, right? So your glucose has to be maintained. If your glucose is higher, glycogen is not broken down. So you have a concentration which takes care of the control, or concentration which takes care of the whole system. This is called feedback mechanism. Similarly, let's say something is attaching to the glucose. So you also need to take care of the concentration of that, because that also impacts the concentration of glucose. So similarly, what I mean here is, in this whole cyclin CDK activity, it's not just the cyclin and the CDKs. There is also something called CDK inhibitors that also takes place, because CDKs also need to be regulated. So there are so many concepts and so many names that you will keep coming across. Remember the core concept, and then try to plug in these information. They will all make sense. Of course, you need to remember the nitty-gritties. For example, which CDC, which CDK, which stage. That's what you need to remember. But the core idea remains the same. So you will come across terms like CKIs, which are CDK inhibitors. For example, P21 and P27. Now, what are the CDK inhibitors? They are basically CDK cyclin complex binders. So they bind to the complex itself, and then they block the activity, right? So all of these, so you might ask, why are they needed to stop the checkpoints? They are needed for the whole idea of regulation. If your cells at this phase is not correct, then if they are present, cyclin CDK2 will form. then the cell might enter S phase. So to stop their activity, you have these different proteins which come and attach to this complex itself. And then you also have several other components which make sure that cyclin E doesn't produce in excess. So all these are taking place in all, so the cell is making its maximum effort so that the cell doesn't undergo any problematic stage. Okay, so that's what's happening. Right. So let's try to understand this concept on this slide. If I did not discuss this in much, but it's the same thing. Like I mentioned, you have CDC, you have retinoblastom, so you have E2F. All these are different accessory proteins which help in the functioning of the CDK cycling. You see these phosphate groups, these when they are attached, they are inactive, when they are removed, they are active. So there is a phosphorylation that you need and a phosphorylation that you don't need. So both of them together, like both of them have to take place so that you can have the active CDK cycling complex, right? We'll try to answer questions so that this becomes easier. If you have any doubt, please let me know. Okay, so let's start with some questions so that we actually get into the whole idea of this, right? We'll also find out who found these out, actually, who found out the CDKs and cyclins. Okay, so the master regulator of cell cycle is which of these options? See, the key CDK in budding yeast is which of these? Nobel Prize for Cell Cycle Control, who found out this ones. And like I said, CDC, CDKs, these come from the system that you are working. So if your yeast has CDC2, which is a human homologue, and these questions are from the uh, lecture notes, so it might be a good revision for you. Anyone? CCAC. Okay. Others? CBCA. All right, I'll just check the answers. Anyone else? No one? You can take a guess. It's okay if you get it wrong. It just helps you understand. Maybe you can see where you are. No, it's not getting clear. CCBA. CCBA. Okay, one more minute and then I can just give the answers. Okay. Okay. Then I'll just quickly tell the answers. Yeah. Right. So the master regulator of cell cycle, we already know it's CDK cycling complex. We have discussed this multiple times now. The key CDK in budding yeast is CDC28. Okay. And then the Nobel Prize, this you should remember. So I told you there were 3, 4 experiments that were spoken about in the lectures. One, two of them we discussed. The third one is this. Hardwell, Nurse and Hunt, who found out the cyclin and CDKs, the MPF actually. Okay. And human homologue of yeast CDC2 is CDK1. So like I said, the CDC comes from yeast system, CDK come from human system. Okay. And one important distinction that I would like to point out here is, just in case it missed you, I spoke about budding yeast here, which is CDC28. There are two types of yeast that have been used for studies, which is one is fission yeast and one is budding yeast. Okay, so when you have fission yeast, these are basically the mode of their division. So one divides by fission, one divides by budding, so offshoots. Okay, so when you have fission yeast, that time the cell cycle regulators mostly composed of CDC2. So this CDC2 that we just discussed, this is coming from a fission yeast. And when you talk about budding yeast, which basically reproduces or basically divides by budding, there the core CDK is CDC28. Okay. And both of these do the same activity, CDC28, CDC2. So their human homologue is the same, CDK1, because the homologue is here determined by the activity. Okay. All right. Just be mindful. Next time if it's fission yeast, that CDC2, this yeast and this are not the same, even though the function is same. This is from budding yeast, this is from fission yeast. Okay. All right. Next one. P34 kinase refers to which? So I told you we will keep coming across this P53, P54. So what is their activity actually? So activating phosphorylation of CDK. Which phosphorylation is activating and which one is inhibitory? Just the next question. And what is V1 kinase do? Anyone? B, C, C, C. Okay, all of these are C. I also didn't see the answer. Okay. Others? Anyone else? Anyone? Okay. One more minute and then we go to the answers. Okay, so people have written and just let me give you the answers. Like people have already mentioned, it's BCCC. So P34 kinase, as you can see, the answer is CDK1. And just for explanation, P34 is basically identified in fission yeast, right? So the P34 is actually CDC2 in yeast and CDK1 in yeast. Okay. and it's a catalytic subunit of MPF. Okay, so it's a CDK1. Okay, the next question is which is the inhibitory phosphorylation? Let's just go this way. So inhibitory phosphorylation of CDK1 is basically at tyrosine 15 and threonine 14. Okay, and what it does is if CDK1 remains inactive if there is phosphorylation at tyrosine 15 and threonine 14. Okay, and these are added by V1 kinase. Okay, and what and ultimately do by why are they inhibiting? They are inhibiting because they are blocking ATP and the substrate access, right? So remember tyrosine 14 15 as inhibitory and activating as 161. So when you have threonine 161 which is activated uh, by CAK. So CAK, CDK activating kinase. So because of the addition of that phosphorylation group, it activates the whole CDK cycline complex, right? So what do you need to remember? Which one is inhibitory? Which one is activating? Who adds the phosphoryl group and who added where? Okay. So inhibitory 14-15 by V1 kinase, activating at threonine 161 by CAK. This is what you need to remember. Okay, right. So as I mentioned then, and the last one, as we got from the second question, V1 kinase inhibits CDK. It's a negative regulator. So it phosphorylates the tyrosine 15 and threonine 14, and it keeps it inactive during G2. Okay, let's move to another question. We'll get it more. We'll get as many questions as we can. So CDC 25 now. We spoke about CDC 2. Sorry, we spoke about, where did it go? Previous page, I think. Yeah, we spoke about CDC 2. We spoke about CDC 28. Let's talk about CDC 25 now. What is the CDC 25? Okay. M cycline degradation happens by what component? Right? Someone asked caspase. I don't know if the person is still on the call or not, but I will anyway tell. It's available in recording, so you can refer it back. Cyclin level fluctuates because why? And APC is referred to as which of these components? Hmm. Anyone? A, B, B, D. Okay. Okay, maybe I'll give the answer so that we can move to the next topics. Okay, so let me discuss these answers with you. So CDC 25 is a phosphatase. Okay, what does a phosphatase do? Kinase adds phosphatase removes phosphate groups. Okay, so CDC 25 basically removes the inhibitory phosphatase from CDK1, right? So they are dephosphorylating tyrosine 15 and tyrosine 14, which are added by V1. So V1 adds, CDC 25 removes it. V1 adds when there is no need for the function to happen. CDC 25 comes into play when you need that complex to function again. So CDC 25 is a phosphatase. Okay. So the next concept that we are now speaking about is CDC 25. And which one does it switch from? So CDC 25 works on CDK1. Okay, we are speaking in terms of CDK1 and cyclin B because we are talking about MPF mostly. Okay, Right. Cyclin levels fluctuate because they are degraded by proteasome, which I already discussed with you because of ubiquitin, right? So these cyclins, they have to be selectively destroyed. So for selective promotion to the next stage, and that happens because of ubiquitin proteasome system. Okay, so this was cyclin level fluctuating. Now all these proteasomes, they also have their own complexes for each of these cyclins. For example, M cyclin here is degraded by APC by C complex, right? And we'll see this later. It basically is anaphase promoting complex, and the next C is cyclosome, right? So what did it do? So what does the name suggest? The name suggests that it is anaphase promoting complex, right? So the M cyclin, or basically also called the cyclin B. M cyclin is basically the cyclin in the mitotic phase, and that is when you actually look into the names of it, it is cyclin B. So this cyclin B is supposed to be destroyed so that you can exit from mitosis, which is the second last step of the mitosis, anaphase. So the complex that actually degrades the cyclin B is a anaphase promoting complex by cyclosome. Okay, what they do, they ubiquitinate the cyclin B and securin. Securin is basically the sister chromatid attachment protein. We told, right? We also said that the sister chromatid. So you can see the regulation is not just at the step of cyclin. Every component is attached and every component is regulated together. So not just your cyclin, the separation of the sister chromatid also is taken place by this ubiquitin complex here, right? So securin is also tagged, and then you have these, we will see later that these tagged or ubiquitin tagged components are then degraded by the proteasomes, right? So the question here was, which of these is actually tagging the M cyclin? It's APC by C. Okay. And like I said, ABC adds ubiquitin. So that's why APC is a E3 ubiquitin ligase, which attaches the ubiquitin onto the protein that is supposed to be degraded. Okay. Right. We will see what does E1, E2, 3 means when we go there. Right. Okay, next one. If this looks very overwhelming, it's okay. These questions are for your revision. So when you are attempting to prepare for your exams or revise these topics, you can always come back to these questions and work it out, right? Okay, next one is the G1 checkpoint ensures what? In which phase of the cell cycle does the restriction point occur? DNA damage activates which checkpoint, and Weinert and Harvard demonstrated what? So basically, this is just how do I say? Questions which seem repetitive, but then these are basically different words trying to answer the same questions. We'll do this a bit quickly because we have only 20 minutes remaining, and I also want to do the ubiquitin proteasome system. So I'll just wait one minute in each slide. Okay. Okay, let me give you the answer so that we can go to the proteasome, and I don't want to miss out that topic. You can, in the meanwhile, keep answering. Yes, I can see people are answering in the chat box. Even if you don't type in the chat box, if you are thinking also, that's enough, because then it tells you that you are thinking in the correct direction, right? Okay, so the first question was G1 checkpoint ensures favorable growth condition, because G1 checkpoint basically checks if the cell has adequate nutrients, proper cell size, growth factor signal, and no severe DNA damage, right? So, and the other options, which basically talks about ensures DNA replication, that's for S phase. Spindle also for S phase, chromosome also for replication and metaphase. So these are not for the G1 checkpoints. Okay. Now the restriction point, R point, this is basically the checkpoint that I just mentioned, which is called from the G1 phase, right? So the same checkpoint that I just spoke about, G1 checkpoint, it's called the restriction point. Okay, so restriction point is here. Remember that. The next one was DNA damage. If there is a DNA damage, which all system will get activated? All G1 to S, G2 to M, and spindle checkpoint, because all of them are associated with DNA replication somehow or the other. For example, G1 to S, right? Why does this checkpoint get activated? Because it is it's supposed to promote the cell to go into S phase. So in the case of DNA damage, it prevents the replication of damaged DNA. G2 to M, if there is a DNA damage in the previous step, it prevents mitosis with the damaged DNA. And spindle checkpoint also gets activated, so that because spindle is indirectly affected by chromosome attachment, so this also gets affected, right? Activated. Last question, Wyandt and Hartwell, what did they demonstrate? They demonstrated DNA damage checkpoints. Okay, so what did they study? They studied yeast mutants, and they showed that delay cell cycle progression is there when there is a DNA damage. So whatever we have discussed so far, all these DNA damage things, they came from these experiments. You can see there are multiple experiments and multiple scientists that keep cropping up, and that's very important because they were the ones. Their experiments were the hallmarks of how they found out these different components. Okay, next one. Replication stress primarily activates what? P21. So this is P21. We spoke about P34 already, right? And CKI function by what mechanism? Okay, I have some answers. Let me just quickly move on so that you can see the answers B, C, and C, right? And I'll discuss them quickly. So the replication stress primarily activates ATR dependent checkpoints. Now, what is ATR dependent checkpoint? These are basically, so what is replication stress? When you don't, replication stress means DNA replication problem. So your replication fork doesn't work, or DNA synthesis is not taking place correctly. So all these different type of problems are taking place in your replication step. And then these kind of problems are sensed by ATR. What is ATR? It's basically ATM and RAT3 related kinase. Let's not go into the details of that. But so this checkpoint get activated, and ATR then activates CHK1, which basically leads to inhibition of CDK activity, stabilization of replication forks, and everything else. Now, what I want to highlight here is that for each problem related to damage of any part of the cell. For example, here is the DNA replication. You have a protein that senses it, and then it reactivates several downstream proteins, which makes it the checkpoint, and then they together function to to reverse the damage that has taken place, right? So where does the conversation of previous slide and this slide merge? It basically merges at the point that your DNA problem is sensed by this ATR. Then I just told you it activates CHK1 and all this. Those are all its checkpoints, and together they function to halt the cell cycle, and it doesn't let it progress to the next phase, and it also corrects the damage that has done happened to the DNA, right? Okay. P21 is activated by P53. Now, what is P21? P21 is a CDK inhibitor. What is CDK inhibitor? What is it important? If it is inhibiting CDK, it is basically stalling, right? It is stalling the cell cycle, and see how it connects to the previous question. So this, I told you about DNA damage is activating ATM, ATR, right? So that ATM, ATR is further, I told you that that checkpoint further acts on several protein. One of the protein that checkpoint acts on is on P53, and P53 acts on P21. P21 is a CDK inhibitor. It goes and binds to the CDK and says, don't work, don't take the cell to the next stage. That's how the control takes place, right? So these are cascade, and these are transcription factors. And you will always find that whenever you are studying regulation in any cell biology or biochemistry or any place, there is a cascade, and most often than not, these different components keep cropping up. So it looks overwhelming, but if you practice or if you study more, it becomes the workflow or the flowchart becomes clearer. Okay. Next is CKI, which is CDK dependent kinase, cyclin dependent kinase inhibitors. They function by inhibiting the kinase activity. We know that already, right? Okay. And that's it, right? What, did I miss anything? Yeah. Okay. Next one. Okay. These are some experimental questions from the previous year assignments. Okay. So there are some statements on your slide. Just find out the incorrect ones. Anyone? So these are experimental based questions, and all the discussion that we have had of different kind of yeast mutant, these are based on those. Anybody? No? Okay, I see some answers. I will just quickly go ahead so that you can see. So it's D and B. I think you correctly answered that. I'll quickly tell you what is it about, right? So it's basically in those both the questions here, you have to find the incorrect statements, right? So the three statements which are correct are CDC28 mutant do not cells do not initiate DNA synthesis. Yes, because CDC28 is required for your entering S phase. That's a checkpoint CDK, right? Next, which is correct, is CDC28 mutant cells cannot progress beyond the start point. Exactly, because start is the CDC28 control commitment point. Commitment point basically is when you have entered that particular, so there's a commitment point in every stages, and when a cell passes through that point, it basically means it has to complete that step now for sure. So it's somewhere around the beginning or in the center, where let's say you have entered commitment point, so you now have to complete M phase. That is what it means, right? So it basically says that CDC28 mutant cells cannot progress beyond the start point, because up till the start point it can come, but beyond the start point it needs CDC28 and it doesn't have, so it cannot go beyond it, right? And CDC28 encodes a cyclin dependent kinase. It is the yeast homolog of CDK1. We know that already, right? What is incorrect is it can progress to S phase if it's a mutant cell. We have just discovered it cannot, right? And the next one is so out of this, the incorrect statement is the stationary phase one. I'll tell about the other one first. So CDC2 levels do not fluctuate, do not change during mitosis. CDCs are CDKs, and we know CDKs don't change, right? CDC2 phosphorylation correlate with high mitotic activity. Yes, because dephosphorylated or active form, which are with the required phosphorylation and required dephosphorylation, they are active. And CDC2 has highest kinase activity. Yes, because they are a serine protein kinase activity, so they do add phosphate group to these amino acids. The incorrect statements are CDC2 becomes phosphorylated on entry to stationary phase. That is incorrect. CDC2 or CDQ1 for that matter, regulation here is tied to the cell cycle transitions, okay, and not stationary phase. So in stationary phase, your activities are in general low. Remember, they are stationaries. They don't want to go to any other phase. So that's not the entry to the S phase. It's for the entry to the cell cycles S phases. Okay. Right. In the interest of time, I have several other questions, but I will skip these for now. You can always find these questions in the recording. I have to move to the last topic, which is ubiquitin proteasome complex. But like I say, these are available as recording. So those questions will be there. And you can always go to the YouTube link and solve these questions, right? I will now quickly move to the ubiquitin proteasomes complex and also give a contrast to the apoptosis, like someone asked. I don't know if the person is still on the call or not, but yeah. Right. So let's quickly, we already have an idea of what ubiquitin does, right? Ubiquitin proteasome complex is basically a pathway, or this system is a pathway for selective protein degradation here, especially in terms in regulation for your cyclin concentration, right? The first component here is ubiquitin. This ubiquitin is a small protein. And I told you, right? You tag the protein with ubiquitin. So it's a tag you add on the protein, which is supposed to be destroyed, right? And so what you do is what ubiquitin does is it acts as a molecular tag. And when you have proteins which have with polyubiquitin, so multiple ubiquitin together, they are basically the target for degradation, right? And as you can see here, that there are several steps of ubiquitin mediated degradation. You have E1, E2, E3. Remember, we just spoke about E3 ligase, right? We'll just see what is that. First is E1. E1 is basically activation. So when you have E1, it basically activates ubiquitin. First, the ubiquitin gets activated by ATP. So E1 works on ubiquitin. Then E2 is a conjugation. So your activated ubiquitin is transferred to E2, which is basically a ubiquitin conjugating enzyme. And the E3 is a ligase which attaches that ubiquitin onto the protein. So it's ubiquitin, then the enzyme, then it onto the protein. So that's the E1, E2, E3 that you see here, right? E1, E2, and E3. E1, activation of ubiquitin. E2 is conjugation of ubiquitin to the enzyme. Third is ligation, where you pass it on to the substrate. Right now, what happens? How does this tag gets determined? How does this tag get identified? So the identifier here is proteasome, which is called the 26S proteasome. Right now, this proteasome has 28 core particle. It's a proteolytic chamber, and there are several regulatory protein. What it basically does is it basically sees, okay, there is a ubiquitin tag, so I need to go to that protein and I will use ATP, unfold and translocate and disrupt that protein completely. Okay, so it basically whatever protein is targeted by that ubiquitin here. So your proteasome will come and it will deconjugate. So take out the ubiquitin for recycling, and then it will substrate will go to lysosome, and then it will degrade in that lysosome there. Right? So you have proteasome identifying, decomplexing, putting in a lysosome. Now, how is this different from apoptosis, like one person mentioned? Apoptosis is a cell death procedure. Apoptosis basically means the entire cell will die. Right? What did I just mention here? That in your ubiquitin system, only a particular protein is degraded. When you talk about apoptosis, it's basically your cell is degraded and the cell can die. The cell as a whole can die because of several problems like DNA damage, which is irreparable, or any membrane organelles or any organelle that is broken. So in that case, you need the cell to die off. When the cell dies off, it's apoptosis. And the how it is done by the caspases. I now mentioned about proteasome. In apoptosis, you have this caspase enzyme which breaks down the cells. That's all. So the difference here is between the cell death and the protein death, right? Okay. Now I will give some questions and I have some 6 minutes remaining. So we'll do some questions, but of course the recording is always available so you can check, right? Okay. The first question here is phosphorylation of retinoblastoma leads to retinoblastoma. Remember we saw RB in one of the slides. So retinoblastoma does what? And the second one is which protein complex plays a role in the degradation of cyclin? We know this already, but this is an easy question. Three and one. Yes, absolutely. Correct. See, the second question we already know. The first one is basically talking about phosphorylation of the retinoblastoma. This basically leads to the release of E2F transcription factor, promoting phase entry. Now, what does it do basically? So the retinoblastoma protein is a tumor suppressor. Also, remember all the P21, P34, P53, whatever we have spoken about is a tumor suppressor protein. Most of these are associated with tumor suppressor or cancer-related proteins. Also, you will see these names coming up there also. Okay. Here in this case, retinoblastoma is a tumor suppressor. Okay, and it basically is at the checkpoint of G1 to S. Okay, when so in its hyperphosphorylated form, when it is when it's hypophosphorylated, that time it binds to E2F. Now where does this image evokes? In my slide today you would have seen now, this is probably in the second slide of the cell cyclin complex regulation. You would have seen there that RB binds to E2F, and then together they prevent transcription of S phase genes, right? But when you phosphorylate it, when you put a phosphorylate on the retinoblastoma, then these S phase genes get activated and then the S phase entry takes place, right? Okay. And the next question we already have discussed is APCC complex is responsible for M cyclin degradation. Okay. Now we'll do as many as we can till our time permits. Okay, I don't want to hold you beyond 8 o'clock. So what is the primary function of the ubiquitin proteasome system if you have to define it? And what is the role of E3 ubiquitin ligase in this whole process? Easy question we have just discussed. Three and four, 3 and 3. Okay. 3 and 3. Okay. So the primary function of the ubiquitin proteasome is to regulate the level of cyclin that we know already, right? By marking the ones which are not required for degradation. The role of ubiquitin 3 ligase is actually recognizing and binds to the target protein. It's basically, so it degrades ubiquitin protein? No, it first adds the ubiquitin. Degradation is by the proteasome complex. Remember, E1 is ubiquitin activation, E2 is conjugation, E3 is subject transfer to this substrate. So basically the third point here talks about recognizes and binds to the specific target protein. So target protein is a substrate here. Okay, right. Okay, moving on. So ubiquitin is attached to proteins via which bond? This we did not discuss, so it's a good thing. This is easy, which is the correct order. Proteins tagged by polyubiquitin are degraded by, this is also very easy, and proteasomal degradation requires which currency, which is also very easy. So you see the topic, once it becomes clearer, it is not that difficult to understand. Anyone? Okay, I will show the answers because it's not that difficult. So the binding of ubiquitin is via isopeptide bonds. This one we know already, E1, E2, E3. We just discussed that if they are tagged by polyubiquitin, you have proteasome that identifies and degrades the protein. And the enzyme required or the energy currency required is ATP. Okay, this you need to remember. Okay, quickly, I think this is the second last slide. So APC we have already discussed, right? Mainly regulates which entry stage, which stage entry. Sumo, oh, there are two, three, I don't know why. Sumo modification generally does. So what is sumo modification? APC activation leads to degradation. This we have already discussed once, so we know that. And cell cycle is unidirectional mainly due to which? This is also very easy if you think about it. Okay. In the interest of time, I will just quickly show you the answers so that I can discuss them. So like we are already seen, APC basically regulates M phase exit. APC, anaphase promoting complex. We know that I told you it comes towards the play at the end of the M phase, right? Sumo is basically a small ubiquitin-like modifier. Now, ubiquitin tags a protein for degradation, but sumo doesn't do that. Sumo, it basically is for regulation signal. It alters the protein interaction. So if it binds to the protein, it will not let it interact. It will change its localization, but it will not degrade it. Okay. APC activation leads to the degradation of cyclin B, securin. We have discussed it. M cyclin and the. Yeah, I'm sorry. Yeah, and cell cycle is unidirectional mainly due to irreversible proteolysis. So when the ubiquitin is degrading the cyclins are gone. So the cyclins cannot come back. That's why you don't have the cyclin cell cycle step going behind. Right? That's what it means. And why? Because they are using ATP. It's not a reversible process. You're using energy currency. You are committing towards it, and your cyclin is getting degraded. You cannot run this cell cycle without cyclin, right? And this is the last question set. We are already 8 o'clock beyond it. So I will just quickly tell you the answers, but when you are trying to attempt, you can just do it at your own end again, right? So loss of checkpoint control leads to which of these? CDK activity is regulated by all except which ones? Budding yeast, I told you fission is and budding yeast is conserved because, and the most critical feature of cell cycle regulation is what, right? So the answer is this, right? So your loss of checkpoint leads to genomic instability. That is correct, right? Because your DNA damage is not controlled and it doesn't get repaired. CDK activity is regulated by all of these except your transcription of CDK, right? And what does it mean? That transcription in any sense would mean that there is increase in protein concentration, right? CDKs remain constant throughout a cell cycle. You don't need transcription of CDKs to enhance the activity of the CDK. That's why everything else, which basically converts cyclin binding, phosphorylation, CKIs, yes, but transcription doesn't matter, right? Okay, and the next one is budding yeast cell control is conserved because core regulators are evolutionary conserved. We just saw CDC25 has a homologue in human, CDC2 has a homologue in human. So they are all functioning similarly, and that's why this is evolutionary conserved. So what it means is if you study a particular protein combination in each system, it is directly translated to human system because they are evolutionary conserved, right? Okay, and the last one is most critical feature of cell cycle is accuracy and fidelity, which basically means that every point in cell cycle is highly accurate. They cannot go haywire. Speed doesn't matter. It can go slow. Energy efficiency is okay. It can halt and get more ATP. There is symmetry, doesn't even matter, but you need accuracy. You need to be sure that the DNA is correct, only then I go ahead. You need to be sure that segregation of chromosomes takes place correctly. You need to be sure that the organelles are there in place. So accuracy and fidelity makes sense the most in your cell cycle regulation, right? Okay, and that brings an end to this session. Yeah, these will be uploaded as YouTube videos also, and your PPTs will also be uploaded in the folder, so you can check them if you have any doubt. um, Sorry to go over time, but I will try to keep it short in the next session. If you have any doubt, you can put in the chat box or else we can stop for the day. Thank you so much.